Good evening, Sai Vedam.
Good evening, ma'am.
There are others.
Ma'am, I'm not sure.
Okay, let us wait for 5 more minutes and see if someone joins.
Yes, ma'am.
Good evening, everyone.
Good evening, ma'am.
Good evening, ma'am.
Some more to join. We will wait for 2, 3 more minutes and I will continue. Start with the chapter. Hope you have seen whatever I did in the previous class. Were you able to see the recording?
No, ma'am.
Why
I
it
didn't
is?
watch it, ma'am.
Yeah, already it has been put in the group, I believe.
Yes, ma'am.
You have any tests tomorrow?
Yes, ma'am. Ma'am on math.
Yeah, yeah, I understood. We'll wait for 2 more minutes, then we'll start with the session. Okay.
Yes, ma'am.
Good evening, ma'am.
Good evening. Okay. So in the previous class, what were we studying? Anybody who went through the recordings? Anybody? Because previous class, many were absent.
Ma'am, we did eukaryotic cell diagrams of animal and like a plant cell, and then mainly we did the like cell membrane fluid mosaic model.
Yeah, fluid mosaic model we did. And we
then
were
we
discussing
used
regarding,
transport
tell me.
mechanisms.
Yes, we were discussing about the transport mechanism. What are all the transport mechanism we did, Sai Vedant?
Ma'am, we did we did simple diffusion, facilitated diffusion, osmosis, and then after that we did like active transport, ion channels, vascular transport, and then uniport, symport, and antiport, and then we just like saw the explanation. And we have to do vesicular transport.
Yes, yes. So it was just a brief description of what are the types of transport which was there. Now, today we will be discussing about in detail about all those transport, how actually the process of transport takes place in a particular cell. Okay, I hope you are ready with your notes to make a note of the points. Yes?
Yes, ma'am.
Yeah. So let me share my screen. Can you see the shared screen?
Yes, ma'am.
Yes, ma'am.
Yes. Yeah, this was the transport mechanism which I was doing last class. Okay, so I had finished with this. Even this is the one which he was telling. These things I have done. So simple diffusion, I told what it is. The process of movement of molecules from a region of higher concentration to lower concentration is called as diffusion. So this is the diagram of the diffusion which you can see. This is how the diffusion of molecules takes place. Usually diffusion is for solute and gaseous molecules which moves passively. Means it doesn't require any energy. For the movement of molecules here, there is no energy required. Okay, that's why we call it as passive transport. Passive movement in the sense it doesn't require any energy. Next, in the same type, you have something called as facilitated diffusion. Here, I will show you with the diagram, then you will understand it well. See, this is facilitated diffusion. You can see the difference between simple and facilitated. See, simple diffusion, this is the plasma membrane, where the molecules are diffusing from higher concentration to lower concentration. It just moves past through the membrane like this. This we call it as diffusion. But in case of facilitated diffusion, what happens? Here you have a carrier protein. This carrier protein helps in transporting these molecules from higher concentration to lower concentration. So if we compare both, this facilitated diffusion takes place in a faster rate. This simple diffusion takes place in a slower rate. Okay, that is the meaning of facilitated diffusion. Now this is osmosis. Osmosis, we know, this is the diagram which is given for osmosis. Osmosis especially is the movement of water molecules. So we speak about the movement of water molecules from a region of higher concentration to a region of lower concentration across a membrane. See here, moving of water molecules. See, there will be some important terms, right? So here, the water molecule is an important term. Moving from higher to lower concentration is an important term. Then across a semi-permeable membrane is an important term. So osmosis is such a substance where is such a type of movement where water molecules move from higher concentration to lower concentration across such a membrane. Then these are channel proteins or channel ions, means certain ions, like it could be calcium, potassium, sodium, chloride, they move across the proteins, carrier proteins. You can see it here. See, these ligand-gated channels, voltage-gated channels, these I didn't tell because they are too much. It is not required for Olympiad as such. For Olympiad, this is not required. They are still more higher order learning. Then active transport. I think from here I had to do. Oh yeah, I have done even this actually. Yeah, vesicular.
Ma'am, which Olympiad are we like focusing on? Like we have to do.
Sorry.
Ma'am, which Olympiad are we focusing on? Like which one do we have to do? Like participate in?
You're telling me which Olympiad we have to participate?
Yes, ma'am.
In what Olympiad? Science Olympiad and Math Olympiad. You have two, no?
Yes, ma'am.
Yeah, it is you who is going to decide the subject. It is good if you take both. We usually say it as NACJS. For NACJS exam, all these topics are there, which is given. What does NACJS mean? In the sense, National Standard Exam in Junior Science. This is majorly related to science subjects. That is all PCM, PCP. And even basic math is included. So basically, what type of Olympiad you are choosing, in the sense, the name of the exam, it is known as NACJS. Okay, so this is NACJS. Why you have to study this? Why you have to pass this? Because nowadays in JEE, further, if you are a JEE aspirant as well, JEE, you know that there are two types of exams. Yes, no? What are they?
Mains and
Main
advanced.
and advanced.
Very good. You know mains and advanced, and you know how competitive it is. But if you qualify in this exam, you will have even the chance of getting seat in JEE Institute with the marks of Olympiad. Understood? The name of the exam is NACJS. That is National Standard Examination in Junior Science. Here, you will be learning all the portions of 8th, 9th and 10th. This exam is usually conducted below 15 years for below 15 years students, where the subjects like physics, chemistry, biology, and even basic maths, whatever you are learning, that will be there. Am I clear?
Yes, ma'am.
Yes, ma'am.
Yes. Yeah. Today we will be even solving some questions with respect to Olympiad. Olympiad questions will be solving. Let me complete the syllabus. In the last 10-15 minutes, we will solve even some of the questions, MCQ questions. Okay?
Yes, ma'am.
Yeah. See, actually, why I am moving so fast for these transport, in the sense, because I have already finished it previously in the previous class. We have to move further, because weekly only we'll get only 2 hours of class, and I have to complete so much of syllabus. That is why. Okay. So here, active transport, in the sense, see, there will be movement of molecules from a region of lower concentration to higher concentration. Usually, what happens? The movement takes place from higher concentration to lower concentration. Here in the diagram, if you see these Hexagonal structure is sodium. See, sodium in this side, inside, it is low concentration. There are only 2 or 3. But here it is high concentration. So from here, the sodium is going to outer surface. See, extracellular space means outside. Cytoplasm is inside. So this is inside the cell. This is the boundary plasma membrane, and this is outside the cell. So outside the cell, the concentration of sodium is more. Inside the cell, the sodium concentration is less. But usually previously you saw that the molecules were moving from higher concentration to lower concentration. There it didn't require any energy. But here what is happening is it is moving from lower concentration to higher concentration. See, since from lower to higher concentration it is moving, it requires some kind of force which pulls it or drags it from low concentration to high concentration. So you can see here ATP is used. The ATP is used and this phosphate is broken here. That broken phosphate liberates energy which is used in transporting the sodium to outer extracellular space. So this is ADP, used ATP is converted into ADP. And here in exchange to it, see here potassium is high in concentration here inside, but it is low in concentration outside. But movement of potassium is reverse. It is like an exchange. For every 3 atoms of sodium, there will be 2 atoms of potassium exchange. See, here it is 1, 2, 3, 3 molecules of sodium. Here it is 2 molecules of potassium. Potassium in extracellular matrix or extracellular space, it is less. But in the cytoplasm, it is more. So from space, extracellular space, it is moving into cytoplasm. Okay, during this exchange, there is ATP which is required for this kind of transport. We call it as sodium and potassium channel. It is a very famous example. And what type of question can come here? They can ask for every 3 molecules of sodium, 2 molecules of potassium is exchanged, or what it is. Every 2 molecules of sodium, 3 molecules of potassium is exchanged. How it is? How many molecules of potassium and sodium are exchanged? That could be a question here, children. So that is why I am stressing it so much. Clear?
Yes, ma'am.
Yeah. So this we have done. Moving to next, to vesicular transport. Meaning what? It forms a kind of vesicle. Okay. See, there are 2 types of vesicular formation. Vesicle in the sense what? Can you see here the picture? See, as something particle is coming near the cytoplasm, the membrane is forming a circle around it. See, now it is inside a circle. This circle is called as vesicle. What helps in forming this vesicle is plasma membrane. Plasma membrane helps in forming this vesicle. Now you can see that in this first process, the food particle or the solid particle is taken inside. It is a solid particle. The movement of this solid particle is called as phagocytosis. Okay. Then, what is pinocytosis in the sense? Here you can see the fluid molecules. The fluid molecule is going inside the vesicle which is formed by the plasma membrane, and it is forming a vesicle like this. The movement of this fluid molecules inside a vesicle, we call it as pinocytosis. If there are any receptors, what is receptor in the sense? They are a kind of protein which act like an antenna. What did I say? Receptors are kind of proteins which are present on the cell membrane. See, previously I was telling about glycoproteins and glycolipids. See, that is what you were not there when I did the structure of plasma membrane. I think Sai Vedant remembers it. What do you mean by glycoprotein and glycolipid, Sai Vedant? What do they act as?
I think it's somewhere in my notes now. Okay.
Ma'am, glycoprotein and I think they are like ma'am, I think they're in the this thing on the cell wall. They help in forming the cell wall for gram-negative and gram-positive materials.
They are not in cell. Where they are present?
Ma'am, glycoproteins are cell markers and glycolipids. They are like help with adhesion.
Both glycoprotein and glycolipid, they act as cell markers. Okay, in prokaryotic cell, you would have written as cell adhesion. Okay, so here, such kind of proteins are present on the surface. We do call that proteins as markers. So if I have to show you, because majority were in there, I will show you it. See, can you see the picture of cell membrane here, children?
Yes, ma'am.
Yeah, the short chain of carbohydrate which you can see here, it has been labeled as carbohydrate. No, this carbohydrate chain, since it is on this protein, see, blue color is a protein. Since it is on this protein, we call it as glycoprotein. Thus, this green color carbohydrate chain, since they are on the lipids, see this yellow or orange color round structures are lipids. So this is called as glycolipid. This is glycoprotein. The glyco means carbohydrate, somewhere related to glucose, no, which is belonging to carbohydrate. Since it is attached to a protein molecule, we call this as
Glycoprotein.
glycoprotein. Yes. Then this is called as glycolipid. So here, these glycoprotein and glycolipid, they act as receptors as well sometimes. They do act as cell marker. They do help in adhesion. So all these are the properties of those carbohydrates which are on the surface. So can you see these antenna-like cells which were there in the ultrastructure previously, which I showed? So these are called as receptors. So what happens? The particles which are present in the extracellular matrix, they attach to these receptors, they form the vesicle and they are taken inside the cell. See, this is receptor mediated endocytosis. Understood? See, there are two types now. Now I will explain. So we have two types to know here. The first one, see, you have a, consider this is a cell. This is a cell. If I elaborate this part, so this is the membrane of the cell which is present. Okay. So consider here there is a food particle. If it is a single cellular organism I am talking. Okay. So what happens as it comes near this cell membrane, takes it inside and makes a depression like this. And it is taken inside and it goes inside like this. See,
And it forms the cleavage.
yeah. It is forming a cleavage like thing, and later what happens? It forms a vesicle inside the cell like this. And this particle is inside the vesicle. This particle is inside the vesicle like this. Okay. Right, I will do it in one complete cell. Then you so consider this to be a cell. Okay. Now what is happening? Food has come here, or any particle has come. The cell membrane has depressed like this, and it is forming a vesicle. Inside the vesicle, this food particle is present. Then what happens? The digestive enzymes which are there in the cytoplasm will dissolve these food particles and breaks it into pieces. Okay, so whatever is required by the cell, this is the nucleus of a cell. Whatever is required by the cell, it will be taken inside the cytoplasm. It will be absorbed by the cytoplasm like this. And whatever the waste substance is there, that fused waste substance will be there, no? That will be thrown out. So how it will be thrown out? Is the problem. Just give me a second. So that will be thrown out like this. Yes. So these waste substance will be thrown out of the cell like this. So the particle is taken inside by the plasma membrane, forming a vesicle. It digests by whatever the nutrients or the enzymes which are there in the cytoplasm move inside, and they digest it like this, and the digested food is taken inside by the cytoplasm, and the waste is thrown out like this. Here you have two processes. One process is where the particle is taken inside the cell. This is inside the cell, no? If the particle is taken inside the cell, this process is called as endocytosis. Did you understand? Did you understand or not?
Yes, ma'am.
Yeah. If the particle is thrown out of the cell here, this process where it is throwing out, outside cell, this process we call it as exocytosis. Exocytosis. Endo is taking inside the cell. Exo is moving outside the cell. Understood? Understood or not?
Yes, ma'am.
Yeah. Yes. So coming back here, I will show you in this picture now. In this picture. Yeah. See. First one is endocytosis. In endocytosis, there are of different types taking inside the cell. It is a process by which the large number of particles are taken with forming the vesicle inside. So here you saw how the vesicle is formed. Based on the type of substance which is taken inside, it can be named as phagocytosis, pinocytosis, and receptor-mediated endocytosis. So here first one, phagocytosis. It is a process in which large number of particles are engulfed in the cells. We do call this process as cell eating the cell. Okay, a large substance is taken inside a cell, that engulfing, capturing it like this inside. Can you see this? See here how this large solid particle is just swallowed by the cell vesicle. So this type we call it as phagocytosis. So next is pinocytosis. See, pinocytosis, it is a process by which large number of particles which are soluble in water are taken into the cell. So this substance which you can see, it is soluble in water, means the fluid content we are talking. That fluid content is taken inside the cell by the formation of this vesicle. So this process of engulfing or intake of the liquid particles, partially soluble particles, we call it as pinocytosis. Next, exocytosis. See, exocytosis I'll show. See, this is receptor mediated, which is not given. See, this is exocytosis. See, can you see the vesicle membrane? See, this is resembling to that of the plasma membrane. Why? Because it is formed by plasma membrane only. Okay. See, exocytosis, the process in which an intracellular vesicle, that is membrane bounded spherical, moves to the plasma membrane and fuses to the substance to the extracellular fluid. See here, I showed the same thing in the OneNote. See, this is a vesicle with certain particles. This fuses with the membrane of the cell and slowly it pushes the substance outside. See, this is fusing here. It is adjusting itself to it and it is pushing the substance outside. So this type of throwing the substance out, we call it as exocytosis. Now, can you relate this with this, whatever I taught here? So this is endocytosis, exocytosis. In endocytosis, depending on what is the type of the particle, you have of 3 types. Did you understand? Did you understand, children?
Yes, ma'am.
Yes, ma'am.
Yeah, please make note of this diagram and write a short statement regarding what is phagocytosis, pinocytosis, and receptor-mediated endocytosis. First, draw this rough sketch. Are you making a note, children?
Yes, ma'am.
Yeah.
Yes, ma'am.
Did you finish?
1 second, ma'am.
No, ma'am.
Yes, ma'am. Done.
Shall I move to the next, children?
No, 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 ma'am. Once again.
Done. Done, children.
Yes, ma'am. Yes, ma'am.
Yeah. Now moving to next.
We're done, ma'am.
Yeah. So this exocytosis also, I hope you understood. Just make a rough sketch of this as well with the definition of what actually exocytosis is. Done?
1 second.
It's okay, take your time.
Done, ma'am.
1 minute, ma'am.
1 minute,
Yeah,
ma'am.
yeah, do it, do it. Did you finish?
I'm almost done.
Done, children? Did you finish?
Yes.
Moving to next.
One second, one second. Yeah, done, ma'am. Now I'm done.
Yeah. Moving to the next one. So next, even this is very simple: uniport, symport, and antiport. These are also the proteins which helps in movement. See, in this diagram, there is simple explanation of uniport, symport, and antiport. Uni means one. Means in this transport system, you can see that there is movement of only one substance in one particular direction. Okay. Only one substance is moving in one particular direction. So this is called as uniport. Symport means two substances are moving in a same direction. You can see the arrow here. There is S1 and S2, which is substance 1, substance 2. They both are moving through the protein channels in the same direction. This is symport. Antiport. Antiport means the substance moving in opposite direction. See here, this substance S1 is moving from outside to inside, S2 is moving from inside to outside. So this is called as antiport. Okay, uniport, symport, antiport. So what are the examples here? See, uniport, movement of single substance, it requires no energy. There is no energy which is required here. The diffusion, facilitated diffusion, all takes place by uniport. So symport, this type. Symport, where two different substances move in the same direction, like glucose transport takes place by this process called as symport. Antiport, just now we studied how the sodium and sodium and potassium channels, how it is moving. Sodium and potassium pump, sodium potassium pump, or sodium bicarbonate pump. All these are examples for antiport. Okay, this requires energy and moving in opposite direction. Sometimes even symport requires energy. So this is the meaning of uniport, synport, and antiport. Any doubts here?
No, ma'am.
No,
Please
ma'am.
take down only this diagram. By this only you can understand. No need of writing everything. Please take down only the diagram. And just in uniport you can write.
Could you just tell again what is antipode and unipode?
Uniport means the movement of substance in only one direction. One substance is moving in one particular direction, that we call it as uniport. Okay. Symport, where there are two different substances, or it could be a same substance but going in a same direction. See the arrow? Same direction it is going, from outside to inside. But anti means against. There are two substances which is moving in opposite direction. That is the meaning of antipode. S1, S2, both are moving in opposite direction. This is antipode. See, write the diagram. Write however it is. For example, if S is there, arrow is shown. Write this complete thing properly. Indicate it with arrow properly, because arrow tells everything. And here below uniport, please give the example here. Diffusion, symport, glucose transport, antiport, it is sodium pump or chloride bicarbonate pump. Write the examples. Co-transport, in the sense there are two transports. Transport of two substances which is happening. So you call it as a co-transport. Please make a note. Are you writing?
Yes, ma'am.
Done now.
Yeah. What about others? Shall I move to the next?
I'm
Yes,
almost
ma'am.
done.
Yes, yes.
I'm done.
Yeah. Now moving to the next. So now we have finished with various types of transport. See, this is one of the major function of plasma membrane, that is the transport of molecules. Okay. See, all these are the functions of plasma membrane, in which transport is very, very important. Now we will move into the next one. We have finished with all these, so I will be moving forward. Eukaryotic cell plasma membrane, cell membrane we have finished. Now coming to the next organelle, which is cell wall. So cell wall is also very, very important. There are various organisms where cell wall is the outermost layer. Can you name some of them, children? Which organism has cell wall as their outermost membrane?
Plants.
Ma'am, plant cell.
Plant cell, yes. Okay. Then what
Algae.
else? Algae is also a plant.
Fungi.
Sorry, your voice is breaking in between.
Mom, is fungi like does fungi have cell wall?
Yes. Yeah, fungi. Fungi also has the outermost layer as cell wall. Then what else? You said plant cell, fungi. The one organism which you have learnt already, that also has no. Which is that which you have already learned.
Mom, bacteria.
Yes, very good. See, bacteria also has the cell wall, which is the outermost covering. Not necessary that bacteria's outermost covering always should be capsule. Okay, some bacteria will have capsule and some bacteria will not have capsule. But their outermost membrane for those which doesn't have capsule will be cell wall. Okay, so here cell wall, you only told me now what are all the organisms. Plant cell, it could be fungi, it could be bacteria. See, is the composition or the chemicals by which this cell wall is made up of in all these different organisms, is it the same?
No, ma'am.
Do you know the composition?
Ma'am, like in myelin sheath, it's 50% protein or like something like that.
No. Cell wall. You are
I'm like
telling
fungi.
about regarding. You are telling the composition of protein and lipid in plasma membrane, Sai Vedant. Don't get confused. I'm talking about cell wall.
Fungi's cell wall is made up of chitin and plants are made up of cellulose.
Very good, very good. Fungi cell wall is made up of chitin, or it is also called as chitin. Chitin or chitin, anyhow you can pronounce it. Then plant cell, you know, cellulose, hemicellulose. Now we are doing with tissues where I have told you that it consists of other depositions like lignin, pectin. Okay, it depends on which type of tissue it is. Okay, cellulose, hemicellulose, pectin, lignin, they are present in plant. Chitin is present in fungi. But what about bacteria? What about bacteria? What is the cell wall composition of bacteria?
Ma'am, glycocalyx and then just two more.
No, no, no. I had told you something regarding gram positive and gram negative.
Ma'am, peptidoglycan.
Yes, very good. It is peptidoglycan. Peptidoglycan is the composition of cell wall in bacteria. So yes, this is required, children. This is very, very much required. And see, one more thing, what is given in the sense. See, here can you see it is given cell wall, a non-living rigid structure called the cell wall, forms an outermost covering for the plasma membrane of fungi and plants. So here, what it is, they can ask you in the exam saying whether cell wall is a living component or non-living component. It is a non-living component. If I take only the cell wall, it is a non-living component. There is only hard deposition of substance. That's it. It is the one which gives shape to the cell. It is like the structural framework to the cell. And it helps in protection from mechanical damage, infection. It also helps in providing cell-to-cell interaction. It is like a barrier for anything which is undesirable macromolecule. Now, we say that plasma membrane is semi-permeable. What is cell wall?
Ma'am, fully permeable.
Yeah, it is permeable. Permeable in the sense it allows everything to pass inside. That is the meaning of permeability. Cell membrane is semi-permeable, means only selected things it sends inside. We also call it as selectively permeable or semi-permeable. But cell wall is permeable. It allows everything to pass through inside. Okay, that is the nature of cell wall. Now, algae have cell wall. See now here composition which is given. Algae has cell wall made up of cellulose, galactins, mannans, and minerals like calcium carbonate. So all these are the components of algal cell wall. While in other plants, it consists of cellulose, hemicellulose, pectin, protein, etc. See, cell wall has different layers. Okay, if the plant is young, it will have only one layer of cell wall, which we call it as primary cell wall. But as it grows, to its inner surface, it adds one more and one more layer of cell wall. We call that as secondary cell wall. I have a picture for this. Can you see this? Can you see this picture, children?
Yes, ma'am.
Yes. So this is what is the layers of cell wall. This is primary cell wall, first form cell wall. To the inner surface, see how many layers are being added. The middle lamellae, it is kind of a glue which helps in attaching to the next cell wall. It is a precursor for the formation of cell wall. So that is made up of, see, middle lamellae is largely mainly made up of calcium pectate, which holds or glues to the different neighboring cell together. Cell wall and middle lamellae may be transversed by plasmodesmata, which connect the cytoplasm of neighboring cells. What is plasmodesmata? I will show you just a second. The cell-to-cell connection is called as plasmodesmatal connection. I don't know whether it is there. Okay, it is not there. So I will show you how it is. See. See, if a cell is like this, it will be cytoplasmically connected to the other cell like this. This is one cell. This is another cell. You could have another cell here which will be connected like this. So it will be a continuous layer of cells like this. This cytoplasmic connections. See, even the cell communicates. See, anything has to be transported across the cell, it moves through these pores, the connections like this. This connection is called as plasmodesmata. The cell-to-cell connection is called as plasmodesmata. This is the connection which will be present between the cells. Understood?
Yes, ma'am.
Yes. Now moving to next. Did you please make a note of the compositions here? Algae. Just write algae. First one, right? Cell wall is a non-living component of a cell. Please make a note.
Done, ma'am.
Cell wall is a non-living component of cell. And what it does, it gives shape, it protects the cell, gives mechanical support, helps in cell-to-cell interaction, and acts like a barrier. All this you can write in one point just by writing one keyword. Is that over? Is it over, children?
Yes, ma'am.
Yes.
Yes.
One second, ma'am. 1 second.
Yeah. If it is done, I will move to the next. Just write the keyword. That's it.
Done, ma'am.
Yeah, moving to next. So even here, give the point as algae and just write the composition. Cell wall, galactons, mannans, and it is made of mineral like calcium carbonate. Other plant cells, cellulose, hemicellulose, pectin, protein. That's it.
Done, ma'am.
Yeah. Moving to next. So this as well. I want you to write this diagram, naming S1, S2, S3, how it is depositing inside. What is S1, S2, S3? Secondary wall 1, secondary wall 2, secondary wall 3. That is how it is. Did you finish?
No, ma'am.
And can you explain middle lamella?
Yes. So this blue color line, what you are seeing is middle lamellae. Middle lamellae, it is made up of a chemical called as calcium pectate. See, it is like, see, I will show you just. It is like a glue in between two cells. Just a second, I will show you. So see, here they have indicated blue color, so even I will take blue color. See, this is middle lamellae, the outermost part of a cell. Made up of calcium pectate. Okay. Next to this, you have the primary wall. Primary wall is here. See, primary wall is the one from which the secondary, tertiary, the first, second, three layers are formed. S1, S2, S3 is formed. See, this primary wall which is present, first is deposits. It deposits the wall which will go, it will be in the innermost one. This is the first form. See here, if it is adding one wall and again adding inside, that one wall will move to the innermost one, no? So S1 is the one which is the innermost cell wall. Like this. Next to S1, you have S2. This is S2. Next to S2, you have, this is S3. Like this, for one cell it is there. In the adjacent, you will have one more cell. For this one more cell, where it went? Here. See, one more cell will be glued like this, which is by middle lamellae. Again, one more cell will be glued here with middle lamellae. And this layers will be there. For these many layers will be there, but when you are writing a cell, how it will be? You will just indicate with two lines. That's it. If this is the plant cell which you are writing, this is the cell membrane, and this will be the cell wall. Just you are indicating it with one thin line here. Since you are studying ultrastructure, this one thin line, if you elaborate, this is how it is. Did you understand?
Yes, ma'am.
Yes. Any doubts here?
No, no, no.
Okay, okay. I hope everyone understood. See, if you have not understood, please do ask like this. I would be able to explain you like that. I hope now it is clear how this wall, various layers are forming. Yeah.
Yes, ma'am.
Yes,
Did
ma'am.
you finish writing? Yes. Did you finish writing? Did you finish?
Yes, ma'am.
So going to the next one. So now we will be starting with various types of cell organelles, starting with the endomembrane system. What do you mean by endomembrane system? In the sense, inside the membrane of the plasma membrane, what are all the coordinated organelles are present, where the function is very much coordinated, we do call it as endomembrane system. So this endomembrane system includes the endoplasmic reticulum, Golgi complex, lysosome, vacuole. See, the functions between all these three will be coordinated. Means what? Whatever the endoplasmic reticular is producing, that will be going to Golgi. From Golgi, it will be transported to through lysosomes and other vesicles, and it could be stored in vacuole. So, like this, the functions are coordinated. That is why this we call it as endomembrane system. You can get a question, children. What are all the various organelles which are included in endomembrane system? What are all the organelles which are included? What are the organelles which are included?
And mom, Golgi,
Golgi,
lysosomes, vacuum.
Golgi complex, endoplasmic reticulum.
Yes, endoplasmic reticulum, Golgi complex, lysosome, vacuole. So apart from leaving this, there are n number of other organelles also, which is very important, like mitochondria, it could be chloroplast, it could be peroxisomes. I will tell you what are peroxisomes and all. Okay, so even though these organelles are present, but they are not coordinated with other components. See, I told you the functions of these four components are coordinated, but these components are not coordinated. Okay, but they are the part which are present within the membrane of the cell. Now, coming first to the endoplasmic reticulum. So this is the structure of endoplasmic reticulum. You can see that always the endoplasmic reticulum is attached to the nucleus. ER, in short, it is called as ER.
And I have to write this much.
No need. No need. I will tell you only which is important. You can write that. Okay. So here, ER divides the intracellular space into two distinct compartments. Means what? I will tell you. See. So if I take an endoplasmic reticulum, consider this is a cell nucleus. So here is endoplasmic reticulum. It is moving like this. And this is the complete cell. Such a huge cell it will be. Okay. So here you can see that it is defining the place which is inside it and outside the cell. The place or the space which is present here. See this space.
this space is called as luminal space. This outside the endoplasmic reticulum in the cytoplasm, it is called as extraluminal. What do we call it as? This is called as luminal space inside the mitochondria. See, this is mitochondrial boundary. Okay, this is inside mitochondria. That is luminal space outside the endoplasmic reticulum. That is in the cytoplasm. This cytoplasm, we call it as extraluminal. Extraluminal. So it is differentiating the cell inner surface into two layers. Means the inside surface of endoplasmic reticulum is called as luminal space. The external surface is called as extraluminal space. Am I clear?
Yes, ma'am.
Yeah. Just a sec.
And
Yeah.
do we write this down?
That is what. Huh?
Do we write this down?
No. No need of writing. So that is what is the meaning of luminal and extraluminal spaces. The ER often shows ribosomes attached to their outer surface. So you can see here, there are two distinct types. One where you can see some dots present on the membrane. We call it as rough endoplasmic reticulum. Since the surface is rough due to the presence of ribosome, we call this as rough endoplasmic reticulum. And here, since the surface is smooth, we call it as smooth endoplasmic reticulum. So the ER bearing ribosome on their surface is called as rough endoplasmic reticulum, in short known as RER. So in absence of ribosome, they appear smooth and they are called as smooth endoplasmic reticulum. Am I clear?
Yes, ma'am.
Did you understand this luminal and extraluminal space?
Yes, ma'am.
Yeah. Take down only this first point, because rest you know. Take down only the first point.
Ma'am.
Yes.
Ma'am, I lost power and disconnected like 5 minutes ago. Can you tell what were you doing?
I was doing with endoplasmic reticulum.
Oh.
Yeah. I was doing with endoplasmic reticulum. I was telling regarding what is this luminal and extraluminal space. Were you there for that?
Yes, ma'am, I was there.
Yeah. After that, I was just telling that yeah, the one which is present, which is with the ribosome, is called as rough endoplasmic reticulum. Without is called as smooth endoplasmic reticulum. And I told to write only this first point because it is not there anywhere.
Okay.
Finished?
No, ma'am.
No, ma'am.
Did you finish?
Once again, ma'am.
No need of writing everything, just the first point you write.
Yes, ma'am, done.
Yes. Moving to next one. So, RER is frequently observed in the cell actively involved in protein synthesis and secretion. So, we know the functions. We have read all these. RER, what is the function of RER? It helps in protein synthesis. So, in wherever there is more requirement of protein, there the RER will be more in number. Could you tell me any example where RER is more?
Ma'am, muscle cells.
Very good. Muscles, proteins are more, required in more quantity. Synthesis is also more because it will be actively undergoing contraction and relaxations. So there in muscle cells, you have more of RER, rough endoplasmic reticulum. Very good. So what is the function of smooth endoplasmic reticulum? It is already given there, which helps in lipid synthesis or steroid synthesis. Yes, tell me.
A detoxification.
Yes, that is also there. Detoxification is also there. So where do you think it will be more in number?
Liver cells, pancreas.
Yes, liver cells, especially in liver cells, they are found in more because their function is detoxification. One of the function of smooth endoplasmic reticulum is detoxification. Means in a toxic substance, there will be one component which will be making it toxic. The removal of it is called as detoxification, and that is a function of smooth endoplasmic reticulum. Since liver is called as the chemical industry of a body, means any chemical, if it is entering into the body, first it will be moving after the digestion, everything, absorption, it goes to liver for distribution. Okay. There, if it finds that it is a toxic substance, there the hepatocyte cells, with the action of smooth endoplasmic reticulum, it tries to makes it non-toxic if it is feasible by the body. Not all toxics are like that, because many toxic, it depends on how impulsively it reacts to the body reacts to it. Okay, so some toxic substance can be made as detoxic by smooth endoplasmic reticulum. So they are extensively and continuously with the outer membrane of the nucleus. Yes, we are talking about rough endoplasmic reticulum. And smooth endoplasmic reticulum, they are the site for lipid synthesis. In animal cells, lipid-like sterol hormones are synthesized by smooth endoplasmic reticulum. So just write as RER, put a hyphen, helps in protein synthesis. SER, put a hyphen, helps in lipid synthesis. Lipid like steroid hormone synthesis, detoxification. Only mention the structure and function. That's it, nothing else. Don't write all the points. I hope you understood.
So which points should we write? Like first to
See, you have to just write RER hyphen helps in protein synthesis. Next, write SER, put a hyphen, helps in lipid synthesis. Then you have lipid-like sterol hormone synthesis and helps in detoxification. In a single line, just give the name of the organelle and tell what it is doing. That's it. Understood?
yes, ma'am.
Wait, ma'am. Lipid, steroidal hormones, and
Synthesis of lipid. See, it is there here only. Comma, lipid-like sterol hormone synthesis.
yes, ma'am.
And detoxification.
Okay, yes, ma'am.
Did you finish?
Yes, ma'am.
Moving to next. So next is Golgi complex. Tell me who discovered Golgi complex?
Camillo Golgi.
Very good. Yes, Camillo Golgi. In the year 1898, he was the one who discovered this organelle called as Golgi complex. See, there are many flat-like structures which are piled up parallel to each other and form a huge stalk like this. These parallel structures, we call it as cisternae. So they consist of many flat disc-shaped sands or cisternaes. These are stalked parallel to each other. See, this is how they are stalked parallel to each other. And they have a particular pattern, okay? It is not like as it is they are forming. They have a particular pattern. Here you can see in this picture, a very well patterned. See, this kind of face, which is a concave face. Sorry, convex face. So this is convex face. This is called as cis face. This is concave face, which is called as trans face. So this is called as forming phase and this is maturing phase as well. See, can you see this vesicle? It is shown as incoming transport vehicle. Where it is coming from?
Ma'am, ER.
Yes, it is coming from ER. It will come from ER. It will join here. In here, it transfers to the cisternaes, and while they are moving, it undergoes various kind of modification, packaging, everything. And through those vesicles, can you see here outgoing secretory vesicles? So through these vesicles, they are being packed and they are carried to the other part of the cell or to outer part of the cell. So what is the function of Golgi in the sense? It helps in modification, transport and packaging. Modification, packing and transport. That is the main function of Golgi. See, the Golgi cisternae are concentrically arranged near the nucleus with distinct convexes or the forming phase. See, this is how it is arranged near the nucleus. Where near the nucleus? Where actually the endoplasmic reticulum is present. Okay, how it is facing? This cis phase is facing towards the endoplasmic reticulum and trans phase usually will be facing towards the plasma membrane. Am I clear?
Ma'am,
See, wait,
I
I
have
will
a doubt.
do it. Just a second. What is the doubt?
Ma'am, how exactly does it modify substance?
Huh? How it modifies the substance?
Yeah, like if in case lipids are transported from ER, how does it modify it?
Not only lipids, even proteins.
Proteins also, yeah.
Yes. See, consider this is nucleus. This is ER. This is ER, which is like this. This is rough ER. And this is smooth ER, which is present like this. It is present. Okay. So along with this, there is presence of Golgi complex here. This is phase like this. And the trans phase is here. So this is how it will be. And the vesicles will be here. So whatever the vesicles are coming out from here, it will join. Consider it is coming from rough endoplasmic reticulum like this, and there are proteins which are inside like this. It comes and joins here in the cis phase. So as it joins to the cis phase, the whatever the proteins which are present inside, it goes here to the cisternae, lumen of the cisternae. Consider it has a protein which is made up of 100 amino acids. What are amino acids? What are amino acids?
Substances which make up proteins in different populations.
These are the individual components which help in formation of protein. Means protein is nothing but a long chain of amino acids. See, consider these small pearls are amino acid. These pearls are linked together. So this long chain of amino acid, now we call it as a protein. Consider like this, it has 100 amino acid linked together forming a protein. But the cell requirement is different. The cell wants two proteins which are having 100 amino acids, which are having 50 amino acids in each. One should have a glucose attached to it. One should have a fructose or any other substance attached to it. So now what it happens, now the protein which is moving here, while it is moving here in the cisternae, it breaks it into two polypeptide chains. By the end it comes there, it adds, for one it adds a glucose chain, and for another it adds a fructose chain. Now whatever the cell requirement was, see, this is what is known as modification. Now it has modified the protein raw protein which it has got from endoplasmic reticulum, and this is the required protein. Now this required protein is transported in this vesicle of the Golgi, and it is, whether it is required for the same cell, it is transported to that particular place, or else it will be moving outside the cell to wherever it is required. That is the meaning of modification, packing in vesicle, and transportation. Did you understand?
Yes, ma'am.
So this is what is the function of Golgi. Any doubts here?
No.
Yes. Going back here. Yes. Coming back here, I want you to write this complete slide with the picture. Please, you have only 5 minutes. Do it fast. finished?
One
No,
second,
ma'am.
ma'am.
Almost.
Did you finish?
Yes, ma'am.
Yes, ma'am.
I'm 1 minute.
Yeah, please finish fast.
Mom, you have to draw it.
Sorry?
And you have to draw that.
Yes. Not exactly how it is, just a rough sketch you make. Especially don't forget the cis phase, trans phase, which is forming phase, incoming phase, outgoing phase.
I'm done.
Yes. Now, I have some questions. I just want you not to answer anything. Just take a sheet of paper and write the answer. At the end, I will give the answer key for it. Did you understand what I said?
Yes, ma'am.
Yes.
Yeah? Just a second. Just give me a second, children. The question looks very simple. Please do answer it. After you finish 1, 2, 3 question, tell me. I will scroll it down. You don't tell the answer in a sheet of paper. Just write what is 1. Only option you write A, B, C, D. 1 is what? 2 is what? 3 is what? You should finish it fast because you have 25 questions. Did you finish 1, 2, 3?
Yes, ma'am.
What about others?
Yes.
No, ma'am.
Ma'am, you can scroll.
Yeah. Did you finish everyone? Moving to next 3. Yes.
Ma'am, 4, 5, 6.
4, 5, 6 is done.
Yes, ma'am.
Next. 7, 8. Just a second. I will. Yeah. 8 options are down as well.
Yes, ma'am, done.
Done. Moving to next.
Ma'am, 1 second, scroll up.
Finished?
I finished.
Yeah. Then you have 9 to 12.
Mom, you can scroll.
Did everyone finish?
Yes, ma'am.
Yeah. Now moving to next. This is primary level. I will give the higher order level in the next class. It looks very easy, these questions for you. Done?
Yes, ma'am.
Moving to next.
Mom, how many questions are there?
You can write only till 20. Another 5 are there actually. You write only till 20 because by the time you correct, it will be some 3-4 minutes.
Okay, yes, ma'am.
Only till 20 you write.
Done now.
Did everyone finish?
Yes, ma'am.
Now please correct it. Now, first one. What is the answer for first one? I will just say the answers first. First one is B. Second one is C. Third one is B. Fourth one is C. Please correct. Are you correcting? You have to tell me the marks at the end. That is why I'm asking you.
Yes, ma'am.
Fourth one is C. Fifth one is B. Sixth one is C. Seventh one is C. Eighth one is B. Ninth is A. 10th is C. 11th is C. 12th is B. 13th is C.
Ma'am, 12th is what?
12th is B. It shrinks due to plasmolysis. Whenever a cell is kept in a plant cell is kept in a salt concentrated.
Oh, sorry, but I'll have to leave the meeting, ma'am.
Sorry?
I have to leave the meeting.
What?
I'll have to leave the meeting right now, ma'am.
So this is B. Next 13 is C. 14 is D. 15 is B. 16 is C. 17 is A. 18 is B. 19, 20. It is C. Did you finish?
Yes.
Yes, ma'am. My God.
Just a second. Just a second. I will stop sharing. Now tell me one by one. Yes. How many are there? Only
5
five, no?
people.
Yes. Sai Vedant,
20 out of 20.
20. Very good. Vamshi,
Ma'am, 20.
huh?
Ma'am, 20. 20 out of
20. Very nice. Then Adarsh,
Ma'am, 19 on 20.
19. Where you lost that plasmolysis?
No, ma'am, the first one, the scientist name.
Oh. Darshita,
20.
very good. Sharanya,
Ma'am, Tarun. Ma'am, 19.
Tarun, is it?
Yes, ma'am.
19. Okay. So yeah. See, these were very easy questions. From next time, I'll make sure that the question is very difficult. Okay. So I hope have you understood whatever we did today.
Yes, ma'am.
Where is Mihir?
Ma'am, he had to leave the meeting, ma'am. He said.
Oh, okay. Okay. Mihir, when did he leave?
Ma'am, like 1 minute ago.
Yes, ma'am.
Okay. Mihir didn't take the ICP test. Okay, fine. So I will conclude the class here. Any doubts in whatever we did today, children?
No,
No.
ma'am.
Please revise whatever we did. Next, we will continue this chapter and hopefully I will finish off by next class. Next class, I will finish off and I'll move to the next chapter. Okay. Thank you and all the best for your tomorrow's test. Perform well.
Okay.
Thank you, ma'am.
Thank you.
Thank you, ma'am.
Sai,
Thank you,
can I,
ma'am.
Sai, can I have your accelerated bio notebook? Can I come collect it and return it on Monday?
Yes, sure.
A recording has been posted in the group. You can hear to the recording and you can write what is there in it.
Ma'am, better if I have notebook, I think.
Okay, fine. Yeah, however you are comfortable. Okay.
Okay. Thank you,
Can
ma'am.
I like
Okay, thank
come
you.
right now?
Okay, sure.
Right now,
All right.
Wait, I don't know. Maybe tomorrow. I'll give it tomorrow, like morning.
I'm out of station tomorrow.
Oh,
Like I'll just cycle all the way there, and then if you're too lazy to come down, I'll just ring the bell and I'll collect the notebook and I'll run off.
come down. When will you come down?
When do you want me to come down? Like right now, is it possible or what?
Yes, yes, sure.
Okay, coming. Bye.
Bye bye. Yeah, bye, bro.